one-step approach and the two-step approach. In two-step RT PCR, the first step involves the reverse transcription, or RT, of RNA by a reverse transcriptase. This reaction generates the first-strand complementary DNA, or cDNA. cDNA is then transferred to a separate reaction tube for the second step of this process. During the second step, the second-strand cDNA is generated and amplified by a DNA polymerase. So, in two-step RT-PCR, reverse transcription and PCR occur in separate reaction tubes. This approach is used when there's a need to analyze multiple targets from the same RNA sample in separate reactions or to store synthesized cDNA for later use. In the other approach, known as one-step RT-PCR, the reverse transcriptase and DNA polymerase are pre-mixed, which allows the RT and PCR steps to be performed in the same reaction tube. Not only is the one-step RT-PCR approach faster, it also requires less pipetting and minimizes possible contamination, thereby improving data reproducibility. It also enables easier processing of large numbers of samples for high-throughput applications.
Chemiluminescent reactions emit light by means of a chemical trigger. As implemented in the ACL AccuStar, chemiluminescence-based technology yields results with greater sensitivity and a broader working range than fluorescence. ELISA, or latex agglutination immunoassays. In APS testing, the hemosil reagent has magnetic beads coated with the antigen. When combined with the sample to be tested, antibodies present in the sample bind to the beads, creating the initial antigen-antibody complexes. The unwanted residuals are removed by a process known as the magwash. To accomplish this, the magnet attracts the antigen-antibody complexes in the cuvette. The plasma and residuals are then automatically removed. The magnet retracts, releasing the antigen-antibody complexes. A secondary antibody, labeled with isoluminol, is added as the tracer element, which binds specifically to the initial antigen-antibody complexes. The magnet once again attracts the complexes. The unattached isoluminol-labeled tracer is automatically removed, and the magnet retracts, releasing the complexes. The ACL AccuStar cuvette containing the complexes is now ready to be analyzed. The cuvette enters a chamber from which all light is blocked. At this point, two triggers are introduced, an oxidizer and a catalyst, producing the chemiluminescent reaction, which generates a burst of light. The fully automated sequence is completed in less than 30 minutes, as opposed to hours required by other methods.